So I decided for my histology project to do the Giemsa staining method. So first of all, what is the Giemsa staining method? The Giemsa staining method is named after the German chemist Gustav Giemsa for his application of a combination of reagents in demonstrating the presence of parasites in malaria. So the Giemsa staining method is typically used for blood-borne pathogens such as malaria. It belongs to the Romanowski stains, which are neutral stains made from oxidized methylene blue, azure, and eosin Y. This mixture is placed on a slide that is post-fixed with methanol. These stains are applied to blood and bone marrow films. So now, what is the Giemsa staining method exactly? So first, we need to create the Giemsa stain solution. For this, you need to add 3.8 grams of Giemsa powder into 250 milliliters of methanol and heat the solution to 60 degrees Celsius. Add 250 milliliters of glycerin to the solution, filter it, and leave it to stand for 1 to 2 months. Then, after the 1 to 2 months, add 10 milliliters of this stock solution to 80 milliliters of distilled water and 10 milliliters of methanol. Now, the Giemsa staining method has two different staining procedures, a thin film staining and a thick film staining. The first procedure, the thin film staining, you need to spread a thin film of blood on a microscope slide. Dip this smear into pure methanol for fixation and leave to air dry for 30 seconds. Flood the slide with 5% Giemsa stain solution for 20 to 30 minutes and then flush with tap water and leave to dry. The second staining procedure, the thick film staining, you need to spread a thick smear of blood on a microscope slide. Dip the smear into diluted Giemsa stain. Wash the smear by dipping into buffered water or distilled water for 3 to 5 minutes and then leave to dry. So now let's look at a disease that the Giemsa staining method is looking for, malaria. Malaria is a mosquito-borne disease caused by a parasite. When an infected mosquito bites a human, the malaria is transmitted to the human from the mosquito. On the right side, you can see a life cycle of the malaria virus. So the malaria virus typically starts in a mosquito. An infected mosquito bites a person who becomes infected with the malaria virus. A parasite, I should say. The malaria parasite then goes into the infected liver, where it infects red blood cells and is carried throughout the bloodstream. A mosquito will then bite an infected person, becoming infected, and the cycle will start all over again. In 2018 alone, there were 228 million of cases of malaria worldwide. Around 2,000 cases of malaria are diagnosed in the United States every year. Infants, children under the ages of 5, pregnant women, and patients of HIV and AIDS are at higher risk of contracting malaria. Children under the age of 5 also make up the most deaths due to malaria. Symptoms of malaria are shaking, fever, sweating, headache, nausea, vomiting, abdominal pain, diarrhea, muscle pain, anemia, and bloody stools. This next graph shows the malaria deaths in the world from 1990 until 2017. As you can see, a majority of the deaths in malaria happen to those under the age of 5. And it also seems like the amount of deaths, the peak of deaths was in 2004, and now due to better health conditions and better health in Africa and other countries, in 2017, it seems the malaria deaths seem to be going down. This next map shows malaria when it was prevalent in many parts of the world that are now free of it today. As you can see, malaria used to be prevalent in all continents. But now today, it is only prevalent in South America, Africa, Asia, Central America, and the Caribbean. It used to be present in the US and Europe, but now the cases that are there are usually from people who have traveled to the countries that have malaria today. So what exactly does the Giemsa stain? First, blood is extracted from the perceived infected individual. This blood is then spread on a microscope slide and stained with the Giemsa stain. The Giemsa solution is composed of eosin and methylene blue. Eosin stains the parasite nucleus red, and methylene blue stains the cytoplasm blue. So, here is a picture of normal blood cells. This next slide shows malaria stains from the Giemsa staining method, with the thin staining method on the left and the thick staining method on the right. Those areas of dark blotting on the graphs is actually the malaria parasite. This concludes my presentation on the Giemsa staining method. Thank you.